It's a pleasure to be here. And I got to say, I love presenting at this conference because there's such a diversity of perspectives. I mean, if you look at this topic of nutritional genetics today, we've had everything from physician in clinical care, clinical nutritionists, geneticists, biochemists, getting at this topic, and everyone from chronic Lyme disease patients to NBA players. I mean, it's just awesome to come at this from so many different perspectives. And my perspective, as you got a little bit from the bio, is coming from one who leads clinical trials primarily. I got interested in this subject because I noticed, just you probably noticed in clinical care, that there's such heterogeneity in the outcomes of my clinical trials. And I knew there had to be a reason for this. And as we started to learn more and more about genetics and nutritional genetics in particular, it sort of sparked my interest in some research that I'm going to present today. All right, so learning objectives is, we've already had quite a bit of this today, but actually delineate, when we talk about nutritional genetics, both Nutrigenetics and Nutrigenomics, and then sort of give you a primer and overview of the state of the science in these two areas as it pertains to micronutrients. So that's my main area of focus, and it's one of the more actionable areas in this field. And then finally put it all together with some tools and give you an overview of that as well that you can use in clinical practice. The clinical actions are related to those. It's identifying micronutrients that your patients might be at risk of deficiency or just suboptimal levels, and then other micronutrients on the other side of this nutritional genetic coin that can be used to help optimize overall genetic expression. And then again, get into the nitty-gritty on a number of labs and interpretive tools that are out there that you can put into practice. So commercial interests really have nothing to do with this subject, but I do consulting and clinical trials with supplements. So we'll start out sort of with the why of personalized nutrition from my perspective, get into some of the terminology and actually some of the research methods that pertain to nutritional genetics. Again, getting into the state of the science on both sides of this coin as it pertains to micronutrients. And then offer my perspective on what you can do in clinical care in 2018 using what we know, and then tie it all together. So before getting into the why of personalized nutrition, we have to look at the why of personalized medicine. Because at least in our Western medical system, our current research paradigm, a lot of the nutritional genetics and personalized nutrition has been an offshoot of the personalized medicine movement. And this is a situation we're all too familiar with. It doesn't get enough attention, I think, though, that it's a one-size-fits-all when it comes to pharmacotherapy. And what you get is, even for FDA-approved medications that are efficacious, you'll get some patients that benefit from it, a positive effect, others with a null effect, and others yet that have adverse effects. Yet on the aggregate, it's deemed to be efficacious. And this is, you look at data like this from the paper in the journal Nature. It's one of the top scientific journals of imprecision medicine, which is what we're doing today. These numbers needed to treat are striking. So ranging anywhere between 3 and 24 people in this particular graphic. And these are not for medications that are really unusual or rare conditions. This is Cymbalta for depression, Crestor for high cholesterol, Nexium for heartburn. The number needed to treat there was 24. So these are drugs that people were taking, and to be honest, we're about as imprecise as it gets with this. A really interesting paper if you've not seen it. So what the goal was in personalized medicine was to transition from this imprecise one-size-fits-all treatment approach into a much more personalized approach, which includes genetics, which has been sort of the focus, but other types of tissue analysis, such that we would have much more tailored treatments based on the individuality of different people. And the reality of this, though, is that it's not working out that great on the pharmacogenetics side. So at University of Maryland School of Medicine, we've been one of the best funded academic institutions with NIH grants, and we really haven't come up with great precision with this yet from the pharmacotherapy side. Making progress, but there's still more to go. And I actually think we have had a better track record when it comes to personalized nutrition. So if you go back a long time ago to Ayurvedic medicine, about 10,000 years it goes back, you had the 3 different dosha types, and there were very different diets prescribed for each of these. You had Vata, Pitta, and Kapha. So they knew a long time ago that it was not one size fits all in nutrition. Now, well, they had 3 dietary types. There's probably more like 300 or 3,000. But they were onto this concept a very long time ago, as were the Chinese and traditional Chinese medicine, where our center's done a lot of work over the years, had different foods for different types of constitutions, much of which has been validated in our Western science. And even the DRIs, most of us realize this, that they vary based on stage of life, gender, age, a whole bunch of different factors. As do the MyPlate guidelines. So they're starting to come along. They've got a long way to go. It's pretty rudimentary personalization, but talking about personalized based on age, sex, height, weight, and physical activity level. So this is starting to be recognized even in your conventional nutrition circles, as the rest of us look to really push the envelope here. And of course, genetics. So this is a piece of the puzzle, but only one piece. So getting into the terminology. I saw my learning objective slides. There's Nutrigenetics and Nutrigenomics. Okay, these are 2 distinct phenomena, and there's even a journal where we publish some of our data dedicated to this. So this is, if you look at personalized nutrition as a coin, you get 2 sides of this. So Nutrigenomics is the impact that nutrition has on genetic expression. So this is your epigenetic phenomenon we all know about now. So DNA methylation, HDAC inhibition, microRNAs, those types of things. Then the other side of the coin is nutrigenetics, which is the impact that our genetics have on our response to nutrition. Okay, and that's really what I'm going to focus on a little bit more today. And this is a nutrition conference. I don't need to get too much into this. You've already heard a little bit about whole food effects of this, as well as macronutrients. I'm going to focus on the micronutrient perspective of this, because there's really an understanding of 3 core concepts that's required here, and that is a basic nutrition knowledge, basic genetic knowledge, and then basic research methods in this area. Then genetics, you had a wonderful primer this morning, so we don't need to go through all this again, get the information in your slides. But what I do want to drive home are the italicized points, a numbers game here. So you got about 30,000 genes, 6 billion nucleotides, and over 10 million SNPs. So when you start to get into research, you're going to need very small p-values to be meaningful, because there's so much opportunity for chance. All right, so I'm not going to bore you too much with research methods, but it is important to know this when we interpret the studies. So the ultimate goal in nutrigenetics is to determine associations between some form of genetic variant, usually it's SNPs, and a nutritional outcome. And there's 2 main categories here. You get your candidate gene association studies. So these are studies that have a hypothesis and are essentially looking at the relationship between some gene of known importance, or hypothesized importance, and an outcome. Then you have your genome-wide association studies, or GWAS. So these really have no hypothesis, and you scan the whole genome or portions of the genome and look at an outcome. All right? And some things to think about in these studies, so just considerations, or minor allele frequency. You heard a little bit about that this morning. This is just a measure of how common is this variant. Generally speaking, at a population level, the higher the minor allele frequency, the more relevant it is. But if you've got a patient who has a variant with a low minor allele frequency, but with a very big effect size, that's going to be relevant to you in your clinical practice. Then you get the whole thing of not overgeneralizing to populations that haven't been studied. So SNPs express differently in different populations, and we need to keep that in mind. We also need to look at replication. So you'll see in a lot of studies that I present today that replication is a key. This helps account for all the confounding that occurs in the relationship between genes and nutrition. So things like lifestyle, environmental exposures, family structure. I've got some pictures of the older Amish, for those of you who aren't familiar with that, as we've done some studies with them as they provide a really unique population for this type of work. And then, of course, chance. Okay, so the more times these are replicated, the more meaningful it is. So a couple more considerations. if you get into the primary literature and look at some of these nutrigenetics studies, you get these cool-looking regional association plots. What you look for, ideally, are clusters of SNPs like this. It's more compelling evidence that something is there. We call them hits, and it's not just due to chance, as opposed to just sort of your lone associations like this that just might have been sheer chance. And as I mentioned before, only the very small p-values are significant in GWAS. So, in your typical clinical trial observational study, 0.05 is the magic number. A little arbitrary, to be honest with you, but that's sort of where we go. But in genome-wide, statistical significance is 5 times 10 to the negative eighth. So these are just things to keep in mind in this field. Okay, so then the other side of the coin, briefly, with nutrigenomics. This is all about gene expression and suppression. I should say it's very rarely a completely on or off phenomenon. It's really just impacting enzyme function at varying levels. So we talked about what some of the mechanisms are. We'll get into some of that in my presentation. And what's relevant to nutrition is the fact that thousands of different genes are impacted by nutrients. Although where we are right now is that we don't really have the specificity that a nutritional component is going to affect the expression of a single gene without others. So it's often affecting the expression of many genes at once. Which is why in some of the studies you see measures of global methylation. We do that. And sometimes specific genes. We're just not there yet, to have a full understanding of what food and food components do with genetic expression. Okay, so I'm going to move on now to just sort of an overview of the science in nutrigenetics for micronutrients. You'll have the slides. There's a lot of information forthcoming. All the references are provided if you want to dig deeper into any of these studies. But, what other place to start than with folate in this subject? Heard all about its impact on methylation today. We know that low levels of folate can lead to birth defects and a lot of other problems. And like many other nutrients, leafy green vegetables are our best source. You get two forms. Folate is the natural form, and folic acid is synthetic. Most of the biological activity is with the methylfolate. So that's one of the reasons why this MTHFR set of polymorphisms is so important. About 40 of them. So I think a lot of patients will get this and they say they have the MTHFR variant, but there's actually a lot of them, as most of you realize. So why is this one sort of the starting point of this discussion when it comes to micronutrients? Well, it's common. So the heterozygous is about half of the population having one copy of this, and homozygous about a third of the population, depending on the population. So that's pretty common when it comes to these types of variants. It has a strong effect size. So the enzyme for which it encodes, the activity is reduced by 30% for those that are heterozygous and up to 65% in those that are homozygous. So this is a pretty big impact. So a lot of the attention that you see on it is well justified. Now, what I think is really fascinating is that in the last couple years, we've gotten to the point where we've sort of evolved from, well, this sounds good in theory, to actually doing clinical trials that are informed by MTHFR variants. So I've got a few of them here, and you'll notice these are all in the last few years. So very interestingly, there was a study of B vitamin complex as a monotherapy for depression, and for people that had some of these MTHFR variants, and noted reduced homocysteine and reduced depressive symptoms, as a monotherapy, B vitamins. So that was pretty encouraging. Study of folic acid for those with the C677T variant. You may be saying, well, why did they do that, not use methylfolate if the patients would have these problems? But that said, those that had high homocysteine with the variant had reduced stroke risk. Those without the variant did not. Also pretty interesting. And then there's the big question I think we encounter in clinical practice. Well, this sounds great in theory, but is there any difference between folic acid and 5-MTHF? And this study here in the bottom found that there wasn't with regard to miscarriage, which is it's a pretty big outcome. There's a lot of different factors that go into that. But there was a difference in serum folate. So there does seem to be a difference in people that have these variants. Okay, a couple more studies looking at adjuvant folic acid with bipolar disorder treatment. And interestingly, MTHFR variants had no effect on the interaction between folic acid and this medication, but COMT did. So there was a negative interaction between these. So something to keep in mind. It's also one of the key variants that you've heard about today. And then the study on the bottom was interesting. It was a 4-arm study looking at B vitamins, omega-3 fats, versus the combination versus placebo. And they noticed that B vitamin concentrations and homocysteine decreased in all of the MTHFR genotype categories after a year, but those who had the TT variant had less homocysteine decrease. And interestingly, and I'm not sure we know the answer to this yet, was that homocysteine steadily increased over the 5 years in all of the study groups. So whether there's some adaptation or something occurring, we're not sure yet. So that's hot off the presses. This is all like very new science here. Okay. So move on to another B vitamin related in B12. We know low B12 is associated with a host of deleterious outcomes. And among relatively healthy people, lower energy than they would otherwise like. And this is important for all people, but maybe especially for those that are on vegan diets, as the primary sources, as most of us realize, are in animal protein. So this is one of the micronutrients that's had a consistently replicated SNP that seems to have a pretty big impact on concentrations, and that's this FUT2 gene that's involved in B12 absorption. So it sort of makes sense that this variant would impact people's B12 levels. So this lead SNP that you see here on the slide has been replicated all across the world. So this is something that seems to have an impact on people, no matter what their ethnic background. So an important one. And of course, there are others, as you're going to see with a lot of these micronutrients. There are many players that are often involved. All right, so we'll move on to vitamin D, which, as everyone here realizes, is that all the vitamins are important, but vitamin D is uniquely important, given its huge impact on genetic expression and activity in basically every biological system. So the process goes from vitamin D3, typically absorbed on the skin by sun exposure, to 25-hydroxy-D to calcitriol. All right, and that plays a role in some of the genetic variants I'm going to talk about here. So there have been a number of genetic variants that have been identified in the literature. Vitamin D receptor makes sense, and then 2 other genes that are involved in hydroxylation of vitamin D3 into 25-hydroxy-D, and then 25-hydroxy-D into calcitriol. So interestingly, this first one, the CYP2R1, has been consistently associated with lower serum 25-hydroxy-D levels. And this paper that cited here, just published very recently, it was a really well-done systematic review. So that's clear. But interestingly, though, people who have this variant tend to have normal levels of calcitriol. So it's been hypothesized, well, there's other mechanisms involved. And this other CYP gene has been shown to be associated with lower levels of that. So the story here is pretty complex, and we'll look at some genes that are involved in calcium as well play a role. So again, like folate, this is an area where the science is brand new on genetics-informed vitamin D clinical trials. So a couple studies on this slide. So looking at vitamin D3 versus placebo for vitamin D deficiency, as you would expect, 25-hydroxy-D levels increased in the D3 group. No surprise there. But what was a bit of a surprise was that a number of these different variants on vitamin D directly impacting that, as well as the GC gene that influences calcium, had more of an influence on follow-up vitamin D than did patient baseline levels, BMI, or sex. So that's pretty impressive. I'm going to show that there's a real role here. Another study that looked at vitamin D versus calcium versus both versus placebo, looking at recurrent adenomas at a variety of different vitamin D and calcium variants, saw a pretty large reduction for a particular VDR variant with vitamin D supplementation, and there wasn't really in this study any role of of calcium, genetic influences on that. All right, so I'll move on now to some of the antioxidant micronutrients, starting with vitamin C, which is important this year for this time of year for immune function, but oh so much more. Low vitamin C levels, we don't see much scurvy here, fortunately, but we do see a lot of these chronic diseases, and vitamin C plays a pretty important role in these. We can get in ample sources of the diet, your colorful fruits and vegetables. And this is, one of the oldest and most classic sort of manifestations of a genetic variant impacting our nutritional status. So humans, as you realize, are one of the few mammals who don't produce their own vitamin C, our own vitamin C. And this is due to a mutation in the gene that you see here. So beyond that, looking at more relevance today, this gene that you see, this SLC, which codes for vitamin C co-transporters, has been replicated a bunch of times and impacts vitamin C levels. Everything to this point has been SNPs, but there are some insertion and deletion forms of genes that have an impact on micronutrient concentrations, including deletion forms of these glutathione S transferases that had 20% lower vitamin C. So again, it's not all about SNPs. There are other forms of genetic variation that matter. All right. Vitamin E is one that's near and dear to my heart as I did my doctoral work on this. Another antioxidant micronutrient, particularly active in the cell membrane. 8 forms of this. I feel like it's a public service announcement every time I talk about vitamin E. It's not just alpha tocopherol. All these other forms matter too. Associated with a bunch of different chronic diseases. Low levels are, that is, some clinical trials suggesting efficacy for certain things. And we get it due to its fat solubility in nature, knows what it's doing. So it's in nuts and oils and seeds and so on. This is pretty common, actually. This is from a review paper a couple years ago. There are lots of different genes that influence these micronutrients. And what you look for with this are those that seem to influence several. At least that's what I suggest doing. So you'll see some of these, the SCARB1, some of the other genes here, ApoB genes. So there's a lot of them, all right? So I think we keep this in mind that it's rarely about one gene when it comes to micronutrient concentrations. Moving on to a mineral, iron. We heard a little about this in the previous presentation. This is one where too little is a problem, too much is a problem, very clearly. So low levels of iron can lead to anemia, fatigue, impaired cognition. Our research team recently published a clinical trial of a very low dose of iron that helped restore non-anemic iron-deficient women back to where they need to be. I wish I had known about some of these variants at the time. We might have been able to screen on that, which, as we'll see on the next slide. But conversely, higher iron levels can also be a problem, all right, leading to oxidative stress and other issues. So we get it in diet from the heme, more bioavailable form red meat, and non-heme and leafy greens, beans, and so on. So again, this is one where there are genetic variants that influence both directions. So those that influence iron deficiency, you see a couple of them here. Again, biologically plausible mechanisms with the transferrin receptor. These are fairly common, so it's something you might see in a patient who's low energy, low iron levels. They don't seem to go up. They have to take a lot. They get constipation, whatever it might be. You might check for this. And then conversely, you have genes that can be involved in excess. So this one's fairly rare, but has a very strong impact. And the last class of micronutrients that we'll look at are carotenoids. So huge class of fat-soluble nutrients, highly pigmented. They give fruits and vegetables their colors. What you see here are the 6 most common in the human diet, also probably the 6 most commonly studied, 3 of which are provitamin A and 3 of which are non-provitamin A. And while it's not vitamin or mineral and technically necessary for survival, low carotenoids have been consistently associated with increased risk of disease and pretty good clinical trials for a fair amount of carotenoids as well. So dietary sources, as I mentioned, your brightly colored fruits and vegetables, they're in there. That's what gives them their color. And beta-carotene is the one that's been studied really the most over time. And this is one that there's a well-known personalized response. So everyone talks, nearly 25 years later about this New England Journal of Medicine paper where beta-carotene supplementation had very different effects on smokers and non-smokers. So standing to reason that there might be a genetic influence on this as well. And there is. So this is one of the biggies to keep in mind that you might want to look for in genetic testing of patients. This is BCMO1. So again, a plausible mechanism here. It catalyzes the provitamin carotenoids into retinol. And this is one that my dissertation mentor, Luigi Ferrucci, he and his team did a systematic review meta-analysis of populations all across the world. And BCMO1 was consistently associated with beta-carotene concentrations and some other carotenoids as well. So alpha-carotene, lutein, zeaxanthin, which is why I think this is one that you might want to look at in clinical practice. Talk a little bit about some work that we've done on the subject. So you'll see in these papers, usually huge authorship teams because it is so multidisciplinary. I'm not a geneticist or anything close to it, but I know about nutrition interventions and so on. So you get these really, it's fun work, although it's expensive, and huge grants are typically required, as it was for ours. I mentioned that we did some of this work on the old order Amish study population in Lancaster, PA. They were chosen because they are homogeneous in a lot of different ways, including the sort of social and family structure. And the result of that, so it sort of lends itself to very efficient work. I talked about the inefficient clinical trials that we would do a lot of times because you had such heterogeneity. This would allow us to do more with less when it came to our population and budget. So we delivered a standardized diet to the home of the old order Amish participants, which was pretty cool. We did some stakeholder engagement to look out and see, well, what exactly are they eating, and crafted a diet around that, and had really high compliance that was measured by the ultimate gold standard of urinary nutrient excretion. And the goal was to conduct the first GWAS of genetic associations with micronutrient concentrations on a standardized diet. So it hadn't been done before. I mean, different diets is the huge confounder here. So this is a really small sample as far as GWAS goes, less than 500. So we were taking a risk, but it ended up working out okay. And did a blood draw after the standardized diet was completed, and then conducted a GWAS. And we did consider batch in this one. That is really important, actually. So completely agree. So Lycopene was one of the areas where we found some interesting data. So when we think of Lycopene, we often think about prostate cancer and prostate health, and there's some pretty good data for that. But the most compelling data really for Lycopene in the clinical world is with lipid management, actually. So there's a couple good meta-analyses on this. And there had been previous genetic associations with SCARB1. This is one that we saw with vitamin E. So this is one that you really want to keep in mind, I think, also as a potential actionable clinical practice item. And why? It encodes cholesterol membrane transport. So your fat-soluble nutrients are going to be impacted by this. We found what's called a nominal replication of this in our work, so it didn't quite reach that 10 to the negative eighth statistical significance threshold, but it was in the ballpark. We did find this novel association on SETD7. So you see here we have this cluster of hits in the same area. And there's a story here. You look at a lot of these papers, if you want to have a clinical story, and it turns out that SETD7 is anti-apoptotic in prostate cancer cells. So this could potentially be part of the mechanism. Well, I want to emphasize with this that this accounted for less than 10% of the variation in lycopene concentrations. So these don't tell the whole story, these variants. They tell part of it, but not all of it. And we published these data in the Journal Nutrients, if you want to take a look. We also found some interesting data with alpha-carotene, which is sort of overlooked, I think, in the carotenoid world. Actually, it's quite potent inhibition of cancer cells compared to beta-carotene, and reduced risk of cancers and all-cause mortality in epi studies. The previous genetic associations with this was the BCMO1. Again, it's with multiple nutrients, why it's one I think you might want to take a look at. We didn't find that in our study, but we found a few others. So we found this novel association, the capin-2, capin-8 locus. And clinical relevance here is that alpha-carotene had been associated with gastric cancers previously. This capin-2, capin-8 gene is involved in gastric inflammation, so this could be a mediating factor here. We found one of these sort of lone wolf hits on PRKCE. We didn't read into this too much, but it is a gene that's involved in malignant tumor formation. We had one of these in a non-coding area, which was, called junk DNA sometimes. We're starting to realize it's far from junk. There's a lot of communication that's going on that we're really just starting to understand now. And this was published in the journal that I pictured earlier. We've continued this beyond the Amish into some NHANES samples. Really tough to get the data, to say the least. NHANES is, as you know, it's downloadable for everybody, but not the genetic information. They protect it, as they should, very carefully. It just makes it tough to do the work. But we did it, and we found genetic associations with essentially every micronutrient that we looked at in pretty big generalizable sample. So it's candidate gene association studies. We chose those that we thought might have relevance. So there's something here, in short, when it comes to genetic associations with micronutrients. And there's a number that I do think you'll want to keep an eye out on. Okay, so I'm going to look at the other coin here just for a few minutes. Nutrigenomics. So epigenetics is something that has sort of entered the popular consciousness now, and it's empowering, I think, to patients to know that their genetic predisposition is not a predetermination. We saw the agouti mouse in a previous presentation today. We have an identical genotype, vastly different phenotypes. We see it in human beings as well, with identical twins. One on the left was a lifelong smoker, lived sort of a hard life, and it shows. I mean, this is a very real phenomenon. So HDAC inhibition and DNA methylation are what we typically look at in these types of studies. So we've heard plenty about methylation. With HDAC, these are enzymes that remove acetyl groups from the histones that wrap the DNA, and you get this situation here when there's HDAC overactivity, where essentially you get this unwrapping, and problems can result when there's too much of that. But what I want to emphasize is that problems can result when there's too little of it as well. And I think we'll find both with HDAC and certainly you've probably seen with some of your patients with methylation that people know, okay, I've heard about methylation, I know it's important, we just load up on SAMe or folate or whatever it might be, and that can create problems, as we heard earlier this morning. Okay, so same deal with methylation. Either hypo or hyper is problematic. And these are just some mechanisms for cancers. This is not relegated to nutrition by any means. There are many medications for serious conditions that are HDAC inhibitors, DNA demethylating agents, or DNA methylating agents. This is a very real set of mechanisms, and nutrition has a very real set of effects on this. One of the, I think, pretty cool, I love to look to nature for things, and I think one of the cool phenomenon that we see is the queen bee, which is genetically no different than the worker bees, but the exposure to royal jelly, which you can get as a supplement. Not going to have quite a profound effect with you as it does with the queen bee, but it's pretty interesting. It goes to show you the power that this has. Okay, and it's not just royal jelly. There are plenty of foods and nutrients that impact this. So you get your vitamins, minerals. Looking at HDAC, sulforaphane as a potent HDAC inhibitor at very reasonable levels, and things like EGCG, curcumin, quercetin, resveratrol. A lot of these micronutrients that we see as having clinical efficacy and anti-inflammatory effects. And if you want to go upstream, it's quite likely that a lot of this is due to the impact that it has on genetic expression of inflammatory genes and so on. Good review paper here if you want to get into the weeds, but these are the ones you need to know about. These are common supplements. These are common components of healthy foods that people should be eating. DNA methylation, the B vitamins are a classic member of this sort of group of nutrients that can have a positive impact. other vitamins and minerals, choline, we heard a little bit about that earlier. Again, EGCG and the omega-3 fats, these are all relatively healthy, healthful foods. And then SAMe. So SAMe is like the ultimate methyl donor that has clinical effects and things ranging as widely from arthritis to depression and liver health, and most likely due to these methyl donating effects that it has. Okay, so I'm going to offer now yet another perspective on, coming from what I would do with the clinical trials and my, pretty exhaustive search literature of what you can do in clinical care with what we have now. So the first step is think about, all right, what type of patients would you want to think about using these tools? So the first is when someone comes to you. So you get a patient who ordered one of these direct-to-consumer genetic tests that I'll talk about and desires some interpretation as part of their plan. So they may come in and be freaked out because they had an MTHFR variant, or like most people, they'll say, yeah, I got this, and I was like, man, I don't know what to do with all this. Can you help me out with it? So that would be certainly an area where it would be nice to have some tools to be able to use it. But then I'd say more from your direction would be patients that have a strong family history, sort of makes sense. Patients that have complex chronic conditions that you've sort of thrown the kitchen sink at and they still don't get better. Because sometimes there are variants that are precluding clinical success. And then lastly is just the motivated patient that's pursuing optimal health, sort of biohacker types, of which I would consider myself one. I've run all the tests I'm going to present today on myself. But knowing the context is key here, all right? So looking at the different types of genetic testing broadly, starting broadest of all, you would have the whole genome, whereas the name suggests the entire genome is sequenced. And there's a lot of interpretations that's required with this, of course, and you see some of the labs here if you're interested. Saliva sample is what's required. The cost has come down in most amazing fashion. Back in the 21st century, the Human Genome Project was $100 million. Now you can get it for about $500. Most people aren't going to need their whole genome sequenced, but it is possible. What more people are going to do is exome sequencing. This is one of the next-gen technologies, and looks just at the expressed genes. This is only 1% of the genome. Some of the most common ones are 23andMe, Ancestry DNA, and then you get a bunch of interpretive tools that I'll talk about, a number of these today. Really easy to turn this in. How many of you have tested your own with 23andMe or one of these? Yeah. So, wow, most people. I have too. So it's become, it's within reach for most people with regard to the cost, as is the interpretation. So we're at the stage now where this is something a lot of people are doing. Then you have just your individual genes. This is often for monogenic diseases, things like sickle cell anemia, cystic fibrosis, but there are others of interest. So you can go to your sort of conventional labs in this respect, like LabCorp, Quest. They have different variants that they can test. And Quest, I'm not sure if anyone's used this. They have this partnership with IBM Watson where they're going to get into some of the broader panels that other labs do. So things are moving in this area quickly. And it's too quickly to keep up with all the tests. There are a ton out there. There are a ton of interpretive tools that can help you treat your patients, and look at just some of these today. And really, there's a number of different categories. So there are those direct-to-consumer genetic tests that offer no or really very limited interpretation. You're not going to do very much with the 23andMe health interpretation that they provide. Then you get just this vast array of different tools for the direct-to-consumer tests that are out there, some that are available to your patients and others exclusively for healthcare professionals. We'll look at both classes today. And then finally, you have genetic tests that are oriented directly for healthcare professionals, and they'll offer these, what I think are quite useful, functional gene clusters that can help with patient care. Okay, so starting with the direct-to-consumer genetic tests, we all know about these. Most of you have had it done yourself. 23andMe, Ancestry DNA, you get your raw genetic data. The problem is that interpreting the data independently is overwhelming for pretty much everybody. I mean, this, for those that haven't had this, you get this. So you'll have your SNP to the left, chromosome and position on that chromosome, and then the genotype. That's not too bad if you stop there, but there are 600,000 rows of data that you get with this. So, I mean, if you're going to go and look on SNPedia for each one of them, you better have a lot of time on your hands. But there are better ways, and we'll look at some of them. And a good place to start, and I'll start with this from most basic and work way up to the more intricate, those that offer more detail for those that are really interested in this. But genetic gene, I mean, does anyone use that in their own data? Or yeah. So this gives a very broad perspective. It's free, so the price is right. They do suggest a PayPal donation, but you upload the 23andMe genetic data, and they give you these methylation and detoxification profiles. These are actually mine. The left is the methylation, and the way it is green is good. It's like the traffic light. Yellow is caution, and red is bad. So you see a lot of yellow and red on mine. I actually don't really have any discernible problems related to this, and I think you will see this is why you just don't treat the SNPs, or in this case even the clusters of SNPs, unless there are problems. And a lot of times you also want to get blood work to verify a mechanism, and then detox as well. And they offer basic interpretation of this. So this is something that a patient might come in, because it's so easy to do, it's free. A patient might come in with and have some questions. The material is okay, but I'm not sure there's a consensus on a lot of this. Maybe the next step up the ladder would be Promethease, which is also quite affordable. It's $5, where you upload your genetic data, and it gives you interpretation of the most meaningful SNPs. And this, I mentioned SNPedia before, which is a pretty good resource. I think for intellectual curiosity, at the very least, it's got a whole bunch of information there. And Promethease draws its interpretation from the SNPedia database. This gives you an example. There's a lot. This can be somewhat overwhelming too. How many of you have used Promethease? Yeah, okay, again, quite a bit. What I like to do is generate a table and then sort by magnitude, which is the evidence strength, which will, again, this is mine. I'm a male, in case you didn't know. It'll give you this a bunch of this, but you're gonna notice conflicts or contradictions. So again, it's to show you we've got to think about bunches of genes together and how it marries to clinical symptoms and how it marries potentially to blood work as well. Okay, yeah, not 600,000 rows, but there are even from something like this, about 15,000. Really, I would focus on those with magnitude, 2 or 3 or higher for those of you that have used it. Okay. Then we get to the interpretive tool that I think provides the most information based on these direct-to-consumer genetic testing. And I know there's a number of folks here who have done training at MUIH, and there's a class in this, Opus 23. So this is just just such a deep wealth of information for those that haven't used it. So this one it uses a patient's 23andMe or ancestry DNA data, but it's only to licensed healthcare professionals. So you have to take a training with good reason. Your brain would melt if you didn't have some type of training on this. The cost is fairly reasonable, but it gets into just this extraordinary depth of evidence-supported clinical interpretation. So again, we've heard a number of speakers today talk about SNP clusters, not looking at a single SNP. This will get into them when it comes to health concerns, things like methylation, detox, inflammation, various disease states. And then what I think is pretty cool too is natural product and medications to support these SNPs. And I'll show you a couple examples. So this is an example of grouped by different conditions. So things like mind-body, digestion, aging, oncology. So we look at clusters of SNPs that would have a role here. So this is a sort of alternate universe traffic light setup where gray is good, green, or gray is sort of like unknown, green is good, and orange is the problem. We'll look at this and some others. Okay. So I find the pathways interesting. This is what I mean, where it could melt your brain if you weren't careful. So this is the methylation cycle, and you can see here where you get your orange is where it could be problematic, yellow, sort of your caution, and gray, things are looking okay. So it'll bring through, and you can see the various steps here in one nice image and package, I think, to see, okay, well, my patient has some of these problems. Maybe it's because FUT2 variants or something like that. So this is nice. And one of the areas that I'll talk about this in closing that I think, it is fairly actionable would be emotional disorders. So this shows everything with the catecholamines. So everything from, starting with tyrosine or tryptophan intake, getting all the way into dopamine, serotonin, norepinephrine, epinephrine. So the different steps along the way. Again, it's yellow where there are problems, orange where there are quite big problems, and then the nutrients that are needed to get there. So I'd say if you want to make this a focus of your practice, it's a good thing to invest in. If you're going to have a more tangential or just surface level, you probably don't need this, but this does provide a tremendous amount of information. All right, look at two more parts of this. So let's say that a patient comes in with a COMT, some COMT variants, and maybe they got anxiety or something else that suggests, okay, this is actually manifesting in them. It would show the agonist and the antagonists. So someone like that would benefit potentially from magnesium, and we see that in clinical practice too. Tends to help with anxiety. P5P form of vitamin B6, and then you get your antagonists too, which are also equally important to keep in mind. So EGCG in green tea is great. It's well studied for many different conditions, but it's not so great often for patients with COMT variants, where it can actually increase their anxiety, and this is one of the mechanisms why. And last is the agents. So curcumin, really well studied for a variety of different outcomes. You can see here what genes is it, an antagonist, and those are for which it's an agonist. So we can see something like curcumin, we know it's got strong anti-inflammatory effects, so no surprise then, NF-kappa B, COX-2, these types of inflammatory enzymes. Interestingly, there's some data for curcumin for depression and emotional disorders, and it turns out that curcumin is an MAO A and B inhibitor. So this is one of the reasons how it might be working. So really cool just for whatever agent you're interested in testing. Okay, moving away from that, we'll look a little bit at direct-to-consumer genetic testing that offers some useful interpretations. So I think the pathway fit tests are pretty good. Anyone use these at all? Yeah. So the pathway fit is one of the pathway tests that's out there that's pretty good. So it gets into things like eating behavior traits, food reactions, micronutrient needs, about $300 or so. So it's not inexpensive, but still within reach. And Nutrigenomics, you heard Ahmed present earlier. So this is a great test. This is one that's available only to healthcare professionals. And the cost, I'm not sure if this is totally up to date, but it provides a range of what it is. And I think this is great. I mean, tests for about 50 or so genes that have strong evidence for many different health issues. So this is one where it's really like the most impactful genes in a lot of different areas, from nutrient metabolism to weight management, eating habits, and physical activity. So this can be useful for patients with chronic disease, but also sort of your elite athlete, performance-oriented person. And then, many here will have Genova accounts. So there's a lot of these healthcare professional-oriented labs have functional bundles that I think can also be pretty useful. Typically about $500 or so. And there's a variety of different tests depending upon the patient's needs. So those with, potential detoxification issues, will look at clusters of those genes. Patients with cardiovascular issues, you can look at clusters of those genes all the way down the line. So these can be pretty useful. The golden rule that you've heard from a few people, I'll say it again, is not to treat the SNPs in isolation. Here's some other things to consider. I think the medical history. If the patient doesn't have a manifestation of the genetic susceptibility, in past or present, then I'm not sure it always makes sense to treat that. I think having some lab confirmation for the variant can be helpful, not necessary, but helpful. So things like homocysteine, we heard a lot about that today, but lipids, glucose, markers of inflammation, if you can measure those, micronutrient concentrations, that was a focus of my talk. And then you get into your NutriVal types where you get these deep nutritional assessments. And again, just think about the clusters of SNPs. It's really going to be one SNP, and the good news is there's enough tests and tools out there from good labs that realize this fact. And just convey, we all know this, but it's worth emphasizing again that the epigenetic influence is usually more powerful. You do the right things, you can usually overcome a lot of these problems. So not just diet, but physical activity, sleep, stress management, all those types of things. So from my vantage point, what are the clinical needs that are best supported by the data right now? I think one that is very well supported would be micronutrient deficiencies. This is the most straightforward application. You get patients who are low in a micronutrient, and that's not always just serum. You want to test functional, so whether with magnesium, it would be red blood cell magnesium, or for B12, looking at methylmalonic acid, some of these types of things, functional levels of this. And then if they have any symptoms that would suggest that they're low, this is a pretty straightforward application. I've listed some of the more impactful and common genes here and then throughout the presentation. So this is one that I think is pretty actionable. I think that there's pretty good evidence also for depression, anxiety, and some of the emotional disorders. So we don't fully understand the mechanisms of these conditions to begin with, but we do know that neurotransmitters are involved in some capacity, and we have a pretty good understanding of how different SNPs on certain genes impact neurotransmitter production reuptake. And we also know how nutrients impact both the expression of these genes as well as just the enzymes irrespective of the gene. So we've got a lot of tools here, I think. I've listed some of the genes that seem to be players and a number of the nutritional elements from B vitamins, zinc and copper and so on. And I find the Walsh Research Institute treatment protocols to be pretty interesting. So if you have patients that are suffering from depression, anxiety, and so on, these could give you sort of a treatment protocol to use for these patients. Some other areas are autoimmune disease and inflammatory conditions. So we know inflammation is multifactorial. You block one pathway. If the offending agent or agents are still there, it's going to go find another pathway. But we do know a number of key genes and transcription factors. Okay, somebody fainted. Okay. All right. I'm sorry if information overload melted your brain. Almost done. Last slide. So I think with that in mind, with inflammation, we know that there's a lot we can do with this. I think it's pretty interesting that we've got some genes and we've got a bunch of nutritional micronutrients and other agents that can help out with this. And I think another area where we've seen some clinical success, I think the data support it fairly well, are environmental sensitivities and the sequelae of that. So the detoxification genes are pretty clear, and we can support this with a bunch of different nutrients. Sulforaphane is one that I've got an interest in. We're running a clinical trial on Nutrigenetics' Abmacol Immune, looking at detox markers. There are others, N-acetylcysteine, liposomal glutathione. a lot of ways that we can support patients who have issues. And if you think your morning commute is bad, that's Shanghai. I counted, I tried as best I could because it's 31 lanes by my count. So it's not just situations like this that contribute to environmental stressors. A lot of it's in the home, and I'm sure a lot of you realize that. So I've taken a huge interest in this because I think a lot of people are suffering and don't realize it. And quite a bit of that derives from their genes just not functioning, having their detox system not functioning as well. You heard all about methylation this morning, so I won't get into that again. So just to tie it together, final slide. Nutrigenetics and nutrigenomics, they're real, okay? So we know that genes influence our response to different nutritional factors, particularly in the micronutrient area. I think that's the area that's most actionable. It's the clearest. The differences are fairly modest, so it's just a piece of the puzzle, but I think it is an important piece, and I provided a number of different genes that you can look at for people who don't seem to be responding to some of the interventions, and this could be one of the mechanisms why. Nutrigenomics, clearly nutrients impact gene expression, no question about that. It's a little difficult to measure at this point in time. It's fairly expensive, and it's mostly in research. I've included this as an outcome in some of my studies, and it can cost thousands of dollars for one good test. And we don't have that specificity yet. It doesn't mean it's not important. It's hugely important. But it's going to be difficult to target genes, like BRCA1 or 2, something like that. You can't say just eat this and it affects these genes and no others. But I think we're going to learn more as we go on. So last point is that, if you're going to use this in clinical care, I think understanding the pathways is clear. You've had a lot of great presentations today that can help with that. Understanding that these SNPs are best viewed as clusters. And then making sure, are there symptoms that reflect this as manifesting? There's lots of compensations that can occur with different genes, even if someone has a quote-unquote bad SNP or bad SNPs. Running other labs, I think, can be useful. And really just look at the whole treatment goal. And that's it for my presentation. Feel free to email me, and I'll be happy to answer your questions.